In this session, we will explore how DNA and RNA function as the molecular blueprints of life, guiding the synthesis of proteins and ultimately determining the traits of organisms. Let's begin by examining the fundamental building blocks of these molecules. DNA and RNA are both polymers of nucleotides. Each nucleotide contains three components: a nitrogenous base, a 5-carbon sugar, and a phosphate group. When linked together, these nucleotides form a sugar-phosphate backbone, creating a stable chain capable of storing genetic information. There are four main nitrogenous bases in DNA: adenine, thymine, cytosine, and guanine. These bases pair specifically through hydrogen bonds, forming the rungs of the DNA ladder. The unique sequence of these bases encodes genetic instructions, similar to how letters form words in a language. Here we see DNA represented as a polynucleotide chain. Notice how nucleotides are joined by covalent bonds along the sugar-phosphate backbone. This structure provides stability, while the nitrogenous bases project inward to form complementary base pairs essential for information storage. DNA replication ensures that genetic information is faithfully passed on during cell division. This process is semi-conservative. Each new DNA molecule contains one old strand and one newly synthesized strand. In this way, cells preserve the genetic blueprint across generations. Replication begins when the DNA strands separate, and each serves as a template for building a new complementary strand. The accuracy of replication depends on the specific base pairing rules. Adenine pairs with thymine, and cytosine pairs with guanine. This ensures faithful duplication of genetic information. This figure shows the replication process in detail. The parental DNA strands separate, and daughter strands are formed by complementary base pairing. The result is two identical DNA molecules, each with one old strand and one new strand. Replication begins at specific origins of replication along the DNA molecule. At these points, the DNA unwinds to form a bubble, and replication proceeds in both directions. Multiple origins allow the process to move quickly and efficiently. DNA replication occurs in the 5' to 3' direction. One strand, called the leading strand, is synthesized continuously, while the lagging strand is synthesized in fragments called Okazaki fragments. Enzymes such as DNA polymerase and DNA ligase work together to ensure the strands are completed accurately. The flow of genetic information follows the central dogma. DNA is transcribed into RNA, and RNA is translated into protein. Proteins are the molecules that carry out cellular functions and determine traits. This process ensures that the instructions stored in DNA are expressed as functional molecules. DNA serves as the genetic blueprint for life. It carries the instructions that determine the structure and function of every cell. The sequence of bases along the DNA strand forms the code that directs protein synthesis and, ultimately, shapes traits. Genetic information is organized into codons, which are triplets of nucleotides. Each codon specifies a particular amino acid, ensuring that the correct sequence is built during protein synthesis. This precise system allows the DNA code to be faithfully converted into functional proteins. This slide emphasizes DNA's role in providing the template for transcription. Genes within the DNA are transcribed into RNA, which then carries the instructions out of the nucleus. In this way, DNA controls the production of proteins without ever leaving the cell's nucleus. The genetic code can be arranged in a chart showing the first, second, and third bases of each codon. The second base plays a key role in determining the amino acid outcome. This system of codons highlights both the redundancy and accuracy of the genetic code. This figure illustrates how codons are read during the process of translation. Starting from a given base, 3 nucleotides are grouped together to form a codon. Each codon directs the addition of one amino acid to the growing polypeptide chain. Transcription is the process of copying DNA into RNA. It begins when RNA polymerase binds to a promoter sequence on DNA. As the enzyme moves along the template, it links RNA nucleotides together, creating a strand of messenger RNA that reflects the DNA sequence. RNA polymerase is the enzyme responsible for building RNA molecules during transcription. It unwinds the DNA, pairs complementary RNA nucleotides, and joins them into a single strand. Without RNA polymerase, the cell could not transfer genetic information from DNA to RNA. This slide revisits DNA as the original template for transcription. Only one strand, known as the template strand, is used to create RNA. This ensures that the RNA copy carries the correct genetic instructions for protein production. Messenger RNA, or mRNA, is the product of transcription. It carries genetic instructions from DNA in the nucleus to ribosomes in the cytoplasm. Once there, it provides the code for assembling amino acids into proteins. Protein synthesis always begins at a specific start codon, AUG, which codes for the amino acid methionine. This codon sets the reading frame for translation, ensuring that every codon after it is read correctly. Without a proper initiation codon, proteins could not be synthesized accurately. Translation begins with initiation when the ribosome assembles around the messenger RNA. The start codon, AUG, signals methionine as the first amino acid in the sequence. This step ensures the ribosome reads the genetic code in the correct frame. The initiator transfer RNA, or tRNA, plays a critical role at the start of translation. It carries methionine and pairs with the start codon, anchoring the process. Once bound, the ribosome is ready to build the polypeptide chain. During elongation, amino acids are added one at a time to the growing polypeptide chain. Each new codon is recognized by a complementary tRNA, which delivers the correct amino acid. This continues until the ribosome reaches a stop codon, signaling termination. Elongation proceeds in three main steps: codon recognition, peptide bond formation, and translocation. First, a tRNA pairs with the correct codon. Next, the new amino acid is joined to the chain. And finally, the ribosome shifts position to read the next codon. Elongation continues until a stop codon is encountered. At this point, no tRNA matches the codon, and release factors bind instead. The completed polypeptide is released, and the ribosome disassembles, freeing its subunits for reuse. This figure demonstrates the process of elongation visually. It shows codon recognition, peptide bond formation, and translocation, step by step. The cycle repeats until the ribosome reaches a stop codon, completing the translation of the protein. This review highlights the central dogma. DNA is transcribed into RNA, and RNA is translated into protein. Together, these processes ensure that genetic information stored in DNA is expressed as functional molecules. This flow of information is universal across all forms of life. DNA acts as the permanent storehouse of genetic information. Only specific genes are transcribed at a given time, depending on the cell's needs. By regulating which DNA sequences are expressed, cells can respond to changes and maintain homeostasis. Mutations are changes in the nucleotide sequence of DNA. Some mutations are small, affecting just one base pair, while others involve large chromosomal regions. These changes can alter protein structure and function, sometimes causing disease, but also providing variation for evolution. This figure shows examples of point mutations, including substitutions, insertions, and deletions. Substitutions may change one amino acid, while insertions or deletions can shift the entire reading frame. Such mutations can profoundly affect the final protein product. This final slide illustrates how a mutation in the hemoglobin gene leads to sickle cell anemia. A single nucleotide change causes valine to replace glutamic acid in the protein. This small change alters the shape of red blood cells, reducing their oxygen carrying capacity and leading to severe health effects.